contraction of muscle cells generates muscle tension, which provides the force necessary for the muscular system to function. The amount of tension generated depends on such factors as sarcomere length, frequency of stimulation, and the size and number of motor units involved. Sarcomere length determines the degree of overlap between myosin and actin filaments. The more the overlap, the greater the number of crossbridges, the connections between myosin heads and actin. Because tension is generated when myosin heads pull on actin, the amount of tension produced in a muscle fiber depends upon the number of crossbridges formed in a myofibril. As sarcomere length decreases, optimizing the overlap, tension increases. Optimal sarcomere length and overlap produces maximum tension during contraction. Look at the graph depicting length-tension relationships. For a skeletal muscle cell, the optimal sarcomere length is about 2.2 micrometers. At this length, the muscle fiber is stretched just above 100% of its resting length. Significantly understretched or overstretched sarcomeres result in lower tension and weaker muscle contraction. In an understretched sarcomere, the ends of the actin filaments reach beyond the midpoints of the myosin filaments toward myosin heads that face in the wrong direction. This hampers the potential for crossbridges, reduces muscle tension, and weakens contraction force. If the length of a sarcomere shortens further, the thick filaments collide with the Z-discs and distort. Muscle tension and contraction force drop sharply. When the sarcomere is stretched beyond the ideal length, there is less overlap between the myosin and actin filaments. As a result, crossbridge formation is substantially diminished, muscle tension decreases, and contraction weakens. In the extreme case, when a sarcomere is stretched to the point where no overlap exists, crossbridges cannot form, and muscle tension drops to zero. Muscle contraction depends on the activity of groups of muscle cells. A group of muscle cells, controlled by a single neuron, is defined as a motor unit. When a stimulus of sufficient intensity is applied to a muscle through a single neuron, all the cells in the motor unit respond by producing a single contraction called a muscle twitch. The muscle twitch, which consists of several phases, can be recorded in a tracing called a myogram. Immediately after stimulation, there is a very brief period where no contraction is observed. This is the latent period. Although contraction is not evident during the latent period, the muscle is not inactive. Calcium ions are flowing out of the sarcoplasmic reticulum, crossbridges are forming, and elastic components of the muscle are being stretched. The first discernible force of the muscle initiates the contraction period. When the muscle reaches its greatest point of contraction, it enters the relaxation period. Motor unit size varies among different muscles. Muscles involved in fine control, such as those that move the fingers, have small motor units. Postural muscles, or those that move large body parts, such as the quadriceps, have large motor units. The amount of twitch contraction force depends on the size of the motor unit. Large motor units contain more cells and can therefore produce more tension and a greater contraction force. The amount of tension generated from muscle contractions depends on the frequency of stimulation. A single stimulus produces a muscle twitch. When a second stimulus is applied before the muscle has time to completely relax from an initial contraction, it exhibits a stronger contraction. This is called wave summation. Successive stimulations at the same frequency will generate a series of contractions with increasing tension. This produces a sawtooth tracing called unfused tetanus. If the frequency of the stimulations increases to a point where the muscle does not have a chance to partially relax between twitches, the muscle will exhibit a smooth, sustained contraction called complete or fused tetanus.